Thank you very much for the introduction. And yeah, it's great to be here and to see all of you involved, or maybe in the future to be involved with the Oncode Accelerator program. And I'm starting off with a patient as we've started off this session as well. So here we see a picture of a patient that is confronted with the diagnosis of cancer. He asked the question, what can Oncode Accelerator do for me? And well, Oncode Accelerator is ultimately, of course, as we've seen during the event, aimed at improving the treatment for cancer patients. So our mission is to accelerate, to optimize the development of cancer drugs, as you've seen in the previous slides, through collaborations to have an impact on patients, cancer patients of all ages. And Oncode Accelerator consists of a couple of activities, and 3 have already been mentioned earlier during earlier talks: patient cohorts, organoid models, and AI. And I will very briefly touch upon some of the achievements that we've managed to in the last year, in this year. And on patient cohorts, Miriam also already alluded to the fact that we are going to collect real-time data on patients. In fact, since last week, there are about 21 hospitals in the Netherlands participating in the RONDA project, which is a collaboration between IKNL and Performation to exchange data from electronic health records with the patient registry, and to do that automatically, to do that real time. And that's a great achievement. Oncode Accelerator program is supporting that and accelerating the further accumulation of data in that registry. That very much helps also in identifying the right patient groups for which we can develop new cancer therapies. AI, an example that is mentioned here on the slide, is Alpha Bridge, where the Netherlands Cancer Institute has been instrumental in developing a tool, an AI platform, that makes structural biology more accessible, using new innovations like AlphaFold, using AI in identifying the right proteins that we can use as a target for new drugs. And also on organoids, we had a great talk by Jarno from the Princess Máxima Center. And also there is a collaboration with CPBT, which is the Center for Animal-Free Biomedical Translation, to use organoids not only to stratify patients for clinical trials and ultimately also for treatment, like in a companion diagnostic, but also to develop models that we can use in a preclinical phase to predict whether a compound, whether a therapy is safe, whether it's toxic, and specifically on the toxicity, which is a critical point in the drug development process. The collaboration will be very helpful to accelerate the drug development process. And you see on the slide regulatory innovation, because we are developing and implementing new models, new methods, and new technologies. And for that, we of course need to make sure that they are properly standardized, that they are validated, that they are qualified by the regulators. For that reason, we engage on a regular basis with the regulators to make sure that these models can be implemented as well. And on the work streams, on the drug development processes that we've covered, and Debbie gave an excellent example of how that works in biologics. On small molecules, the first collaboration agreements have been signed with big pharma and with biotech company. Unfortunately, I can't disclose the identity due to confidentiality, but it's the first step in further building on the network within the Oncode Accelerator program. And Debbie already alluded to the collaboration with Halix to streamline and innovate manufacturing of antibodies. On cell and gene therapy, it's good to see that The Netherlands Cancer Institute is speeding up the development of a clean room facility that will accelerate the production, the development and production of new cell therapies, and that will be open mid next year. And on therapeutic vaccines, it's great to see that we now have had the opening of a cancer vaccine facility at the Princess Máxima Center in collaboration with the Utrecht University, where new antigens are identified that form the starting point of developing new therapeutic vaccines. So, to sum up with some figures, although we started rather recently with the operational phase, we already have 90 partners that have expressed interest in working with us for demonstrator projects. And demonstrator projects are intended to, as Debbie also explained, to demonstrate the added value that implementing these innovations in these drug development processes can bring. Because we need, of course, to show, to demonstrate the added value of that. And everybody can apply for a demonstrator project. Can be academic institutes, biotech companies, pharma companies from the Netherlands or abroad. We have business developers that roam around in the world looking, attracting companies that want to work with us. And we have co-funding available to execute these demonstrator projects. And as previous speakers have already indicated, science is the fundament of what we do. So already now 19 PhD students, 19 PhD projects have started to further build the science, but also to translate science towards drug development improvement. And already there have been 9 Oncode Accelerator funded publications, some of them in high impact journals. But we're not only focused on the collaboration within the Oncode Accelerator program. We also work closely with other national growth fund initiatives, like Helder Eye, that is setting up a data framework, a data infrastructure for the secondary use, the primary and the secondary use of patient data, where we work closely together also on data in the oncology field. The CPBT was already mentioned to set up tox models and other organoid models, and also to standardize, validate, and qualify it. Pharma NL is looking at what are the gaps in the Dutch ecosystem in terms of drug development, in terms of shared infrastructure, in terms of human capitals. So we work closely together to identify those gaps also in the ecology ecosystem, and try to bring funding to close these gaps. Next Gen High Tech is another growth fund program where new equipment is developed, like organ-on-chip production devices, but also cell production devices. And together with them and with the ICAT, the Innovation Center for Advanced Therapies, that is part of the Regmed XB growth fund program, we use their pilot production facilities and the production equipment that is developed by Next Gen High Tech, by companies in that program, to optimize, to accelerate the production process of cell therapies, which will contribute to the acceleration of new cell therapies and new ATMPs. And finally, Biotech Booster is another example of a growth fund program that is supporting startup companies, which is crucial, as René alluded to support that. So we are also working with them to support any spin-outs, either on technology that is being developed in the program, or new drug candidates that are developed, and to facilitate them to spin it out and to start up their own business. So you've seen this picture before from Debbie, and this is to explain that we are well underway to build bridges between scientific disciplines. People, that's what I already get back now, that people that did not know each other from different disciplines through the Oncode Accelerator program already start working together and seeing the added benefit of connecting. But also, of course, building bridges between preclinical science and patients. So by introducing patient data, patient-derived models in the preclinical development process and using these models and patient data to make the connection to patients. And by building those bridges, we contribute to the vision that we have to develop a world where every cancer patient has access to precision medicine through innovative and integrated R&D ecosystem where collaboration plays a key role. So with that, I would like to conclude my presentation. Thank you.